Hi, I am Sanna. I am a medical student, and I'm going to present my research that talks about interleukins polymorphisms and thyroid cancer. So the incidence of thyroid cancer has increased in the last 4 decades, and among the causes, the most stressful life of the population can be important. Chronic stress can cause cortisol receptor resistance, which affects hypothalamic pituitary adrenal regulation. As a result, there an increase in oxidative stress, chronic low-grade inflammation, and immunosuppression. Our group and others have found elevated levels of interleukin-17, both in serum and the thyroid cancer tissues. Also, interleukin-17A acts in tissue defense and maintenance, and this protein can activate the NF-kappa beta pathway, which is responsible for the decrease in cellular apoptosis and for the damage caused by oxygen-free radicals against DNA. Interleukin-17 has thousands of single nucleotide polymorphisms, which can contribute to tumor aggressiveness and metastasis by modifying the sequence, structure, stability, and function of the corresponding protein in thyroid tumor microenvironment. Thereby, we want to identify possible impacts of single nucleotide variants, or SNPs, on IL17A gene, structure, and sequence. For that, we use PredictSNP, ProVM, PMUT, Multipred, and Align VGD. Of 2,464 SNPs found, only 130 were missense type. Then, 4 SNPs considered deleterious in at least 70% of these terraforms were selected for stability analysis in MuPro and mutant. Then, possible post-translational chains in MuSDIP were investigated. Finally, HOPE provides detailed reports of each variation. The HOPE tool identifies that SNP with final 10 exchange arginine for cysteine at position 95, and this can be deleterious since cysteine is smaller and more hydrophobic than arginine, which can generate loss of external interactions and loss of hydrogen bonds. Also, the charge of both residues is different. The SNP with final 82 exchange leucine for proline at position 139, and this can cause loss of interactions with other molecules and disrupt signal transfer and affect protein function, since proline is smaller than leucine. Multipred predicted that variation can alter transmembrane protein with loss of the catalytic site at position 144 and alter metal binding as well. The SNP with final 28 exchange gluten acid by lysine at position 91. Lysine is bigger than S-glutamic, and that can disrupt interaction with other molecules or other parts of protein, such as hydrogen bonding. Furthermore, the difference in charge will disturb the ionic interaction made by glutamic acid, and Multipred predicted that variation can alter transmembrane protein with loss disulfide bond at position 94 and also loss metal binding. Finally, the SNP with final 58 exchange arginine for histidine at position 123, with histidine being minor and neutral. The difference in size between these residues alter the correct position to make the same hydrogen bond. And also, this variation is in a conserved region. The finds are confirmed in MuPro and iMutant platforms, which identified that these variants can cause a decrease in stability of the analyzed proteins. And MuSDIP found a methylation in arginine in these positions. Unfortunately, the epidemiological data of these SNPs show that all of them have low math, making their population validation difficult, and no description of clinical trials were found in NOMAD. Therefore, Bioinformatic's analysis of total 2,464 IL17 gene identified 4 variants that are likely to play an important role in pathogenesis of thyroid cancer and that may be related to its development and evolution. I want to thank you to all of my group here in Brazil for all the orientation and support. Thank you so much.